Every time your heart beats, it burns a massive amount of energy. To trace where that power comes from, we have to push past the surface tissue and drop down into the raw, contracting muscle fibers. In this electron micrograph, darker shapes packed tightly between striated muscle fibers are mitochondria, stationed exactly where energy is consumed. They easily adapt. Look at the right diagrams. In a sperm cell, mitochondria change shape, wrapping tightly around the tail to propel its movement. These power plants are ubiquitous. Depending on the cell type, mitochondria can occupy up to 20% of its entire internal volume. To generate continuous power on that scale, this organelle relies on a highly specific, semi-autonomous architecture. Mapping those layers reveals exactly how they fuel human life. Looking at this 3D model, the first feature is the double membrane structure. The smooth exterior is the outer membrane, composed of roughly 40% lipids and 60% proteins. Zooming in on the surface, we see that this outer shell is highly porous. It's studded with beta barrel structures called VDACs, voltage-dependent anion channels. These open pores allow ions and small uncharged molecules to flow freely in and out. However, larger molecules, like the complex proteins required to keep the engine running, cannot slip through these VDAC pores. They need a dedicated transport mechanism. This diagram maps that mechanism. In the upper left, we see the TOM complex, the translocase of the outer membrane. It acts like a selective gatekeeper, recognizing specific transit sequences on precursor proteins and actively feeding them through the outer membrane. While the outer membrane provides a structural shell, its primary function is a biochemical gateway, dictating exactly which raw materials enter the power plant. Pushing through that outer shell, we drop into a narrow gap, the intermembrane space. Directly below is the inner membrane, which acts as a strict diffusion barrier. It is completely impermeable to ions without specific transport proteins. Unlike the smooth outer shell, as we see in this cross-section, the inner boundary membrane folds deeply inward. These folds are called cristae, and they exponentially multiply the available surface area for chemical reactions. To maintain this dense architecture, the folds must be anchored. This schematic highlights the mycose complex, specifically the MyC60 and MyC10 subcomplexes, which bridge the boundary membrane and hold the narrow cristae junctions in place. This vast folded surface area is where the actual energy production happens. Here we see the machinery of oxidative phosphorylation. Complexes 1 through 4 form the respiratory chain, pumping protons across the cristae membrane to build up a powerful electrochemical gradient. On the far right of the diagram sits complex V, or ATP synthase. It harnesses that accumulated proton gradient to spin its F1 particles, physically converting ADP into ATP, the universal energy currency of the cell. These intricate, convoluted inner folds are mathematically optimized to pack as many ATP energy mills as possible into a microscopic space. Pushing past that inner membrane, we finally arrive at the deepest chamber of the organelle, the mitochondrial matrix. This space is filled with a dense chemical soup. It maintains a high pH of around 7.9 to 8, which creates the gradient needed to drive ATP synthesis, and it houses all the soluble enzymes required for the Krebs cycle. If we look at the upper right panel of this diagram, we find something unique. The matrix contains its own circular DNA, labeled here as mitochondrial DNA, along with its own specialized ribosomes. Because it possesses this genetic material, the mitochondrion is a semi-autonomous organelle. It translates its own genetics and builds several of its own structural proteins without relying on the host cell's nucleus. This provides compelling evidence for the endosymbiotic theory. The mitochondrion operates with its own blueprint, much like a captured bacterium living perpetually inside our cells. These structures are highly dynamic. Mitochondria constantly fuse together into larger networks and divide apart, adapting to the shifting energy demands of the cell. But there's a fascinating exception. This microscopic image captures an animal called spinal loricus, which completely lacks mitochondria. It survives deep in salt-saturated, lightless ocean basins, completely devoid of oxygen, evolving without this cellular power plant. Bringing it back to the human context, let's review how these components work together. We move from the porous outer membrane and narrow intermembrane space through the strict barrier of the inner membrane, the expansive cristae folds, and finally into the genetic matrix core. For almost every eukaryotic cell on Earth, mastering this precise, multi-layered anatomical architecture is the strict biological prerequisite for sustaining complex life.